So let's start. Okay. Good afternoon, dear colleagues. So it is a pleasure to welcome you all to Photosynthesis from Fundamental Mechanisms to Applications, the satellite meeting of the FSPB Plant Biology Europe Conference that was held in Budapest, and now we are here in Gárdony. As the main organizer, I am delighted to see such a diverse and accomplished group gathered, truly reflecting the vibrant European photosynthesis community dedicated to unraveling its complexities. Photosynthesis, the very basis of life on Earth, continues to present us with profound scientific challenges and unparalleled opportunities. From understanding the fundamental light harvesting and electron transfer processes to optimizing photosynthetic efficiency for sustainable agriculture and bioenergy, the insights we gain from studying it are more important than ever. In an era where global climate change and food security are paramount concerns, our collective research holds immense scientific significance. Over the next few days, we have a very promising program featuring both leading experts and emerging talents presenting their work in talks and posters. I hope this conference will serve as a vital forum for sharing novel discoveries, strengthening collaborative relationships, and expanding our understanding of this fascinating field. During the 4-day program, we will place particular emphasis on photosynthetic light harvesting and electron transport, biogenesis and maintenance of the photosynthetic apparatus, photosynthesis under stress, carbon assimilation and biomass accumulation, improving photosynthesis and applied photosynthesis, and finally, algal biotechnology. Altogether, we will hear 33 talks and 24 posters will be presented. We have 2 poster sessions scheduled, one tomorrow on Monday and another one on Tuesday. Posters will be also accessible, of course, during the coffee and lunch breaks. For those presenting posters, especially our young scientists, this is an excellent opportunity to share their ongoing research and receive valuable feedback and constructive criticism. Additionally, we will also have 3 poster prizes with small gifts, and the awardees will have the opportunity to present their talks in 5 minutes on the last day on Wednesday, before the closing remarks. And then for this, I would like to ask 3 volunteers who could help us to select the most interesting and impactful poster presentations. If you are willing to help me with this, then please approach me during the evening. And then the scientific program of this conference was organized by researchers from the Hanran Biological Research Center in Szeged, Institute of Plant Biology. That was Petar Lambrev, Bettina Ugi, János Györgyei, and myself. From Eötvös Loránd University, we had Katalin Solymosi, and we also had 2 international organizers, Cornelius Vitel from Gothenburg University and Iftak Jacobi from Tel Aviv University. And now I would like to take the opportunity to to thank all the co-organizers for their help. And the technical and administrative duties were expertly handled by Remedicon, specifically by Gábor Cirkus and Monika Ivan. And now I would like to hand the microphone over to Gábor, who will share us a few technical details with us. And if immediately after his announcements, then we will start our first scientific session that will be chaired by Xenia Johnson from Szeged, from Marseille, sorry, originally. Because originally, actually, Imre was supposed to chair this session, but due to family reasons, he couldn't come. So this is why now Xenia is our first chair from Marseille. So this session is a photosynthetic light harvesting and electron transport. So welcome you all, and I wish you a very successful and stimulating meeting. Thank you very much.
The relative importance of monomeric and let's say of different component of the antenna population in the stacking. So we consider the mutant CH1 of Arabidopsis. This is a mutant that is unable to synthesize chlorophyll B. That is an important chlorophyll necessary for the folding of the light harvesting complexes. Indeed, this mutant without chlorophyll B, there is a very small antenna size, essentially because the antenna protein are unstable and get degraded. With the exception of CP26, that is the only
dear conference president, dear chairman, dear colleagues. First, I want to thank the organizer that they allow me to present our recent result in this conference in this nice place. My name is Dušan Mazar, and I'm from Czech Republic, from Palacký University. And the results I will present to you are collaborative work of several authors within our consortium. The people are described here, and also the other person participating. So I will tell you first some introduction about oscillating light and oscillations and such.
Structured information, where of course with the coming up with the cloud EM, we have loads of them floating around nowadays, and of course the theory. So to put the question more simply, what we want to do is, for example, this up to 400 molecules, right? You can think of them as 400 energy states. What we want is to lay out in a matrix form 400 states, 400 states, and connect them.
I believe that you must, during your excitation, you must also hit the CP47, right?
Yes, in a sense, yeah. Yeah, true.
So there should be some signal from CP47.
So the CP47, right, is assumed that the core complex itself, everything that, yes, I dissect one CP47, but within 10 picosecond, everything will be down to some some terminal state and stays there. So that is kind of like given already. So within 10 to 20 picosecond, whatever you excite goes all the way down to the terminal states.
Okay.
Then after that, that's really the inference of the transfer.
Okay, got it. So, questions?
Is ubiquitous, highly conserved among photosynthetic organisms in terms of structure and also function, being an essential component of the photosynthetic apparatus. But nevertheless, it does have quite a bit of diversity with respect to, for example, the oligomeric state or some smaller subunits, or especially because of its ability to adapt to find many different types of peripheral light harvesting antenna. Then it can form various super complexes of the core and different antennas that the different organisms have evolved and adapted to their environment.
What structurally gives you that sort of functional role, having that prominent position of energy transfer? Your heavy lines here that you see from the far red chlorophyll dimers.
If I understand you, you're asking what is the basis for the fast energy transfer?
Yeah, structurally.
It is the way that we calculate energy transfer. We only
10 is one.
Yeah, we did not measure quantum.
You did not measure.
No. Only the bryopsis is much more stable, and we have a higher resolution structure, so that and I only show bryopsis.
No, because the maybe one interesting aspect is that for bryopsis you have the PSAMs, which are administered, so that would be interesting to see actually with this.
Yeah, we don't have quantum models for the moment.
Because right now, I don't think you could apply the nanosensors across the population diversity. But that's the only reason. So if you've identified which genotypes of the conditions are different, then I think it's very useful. But first, let's identify, we're flipping it around, say identify where something is changing.
But there's actually also this 1,000 generation of
Yeah, and because it is wood, so it's one cutting in these kind of things. Which we are very far from understanding what's going on in these things. I mean, it's already, visual grade signaling is already strong. So that is because that you will also have ongoing stations. But okay, that is only on the which could also influence.
Yeah, actually, so it talks around 40 kilodaltons, we believe that explanation. So the Grana stacks, the diffusion of the reduced plastopin in the Grana stack is very low, and actually the diffusion of the complexes too. So the idea, the hypothesis behind this is that when you have a stack, you increase that diffusion of the plastopin pool, and also the diffusion of the complexes in the membrane. So that will facilitate the electron sync flow. So it has been shown in a paper a couple of years ago, that actually it's located on a stromal side of the thylakoid lamina. Not in the thylakoid line.
Photosystem 1, photosystem 2. There's a change in the ratio.
Yes, I mean, I didn't show it because it's very preliminary results. It seems that the double mutants have slightly more PS1 than PS2 compared to the wild type. Yeah, I do have to repeat it because I don't have enough experiments to do, but it seems that they rather accommodate more LHCA, perhaps, and slightly more PS1. Still, like when I show these experiments here with the nicomycin chloral, here, despite the fact that they are completely dark green, here when I did the BCS measurements, I couldn't detect any PS2, PS1.
Yeah, that was just a question.
Like the whole active reaction centers were done, so they are doing nothing. Yeah, it's just like the nucleus doesn't realize that they should not do
That's
more.
interesting. The other question was with your LNTP1, if you tried in vitro assay, if you can accelerate photoactivation in the assembly of the manganese cluster.
Yes, so in fact, TLP 18.3, even though that is not published in the TLP 18.3 papers, I did